Well, hello everyone, and welcome, and thank you for joining us today. I'm Dr. Carlos Bosques, CEO at FOSSI, and it's an honor to welcome you to our last scientific discussion of our seminar series for this year. At FOSSI, our focus is to understand, translate, and intervene with food allergies. And over the last year, we've been having a lot of great discussions with experts on the field on the topic of the impact of food chemicals in food, the chemicals in food and the environment on human health. Especially one of the key questions that FOSSI is interested and has been working on is why has there been such a rapid increase in food allergies over the last few decades? And in September, we introduced this new seminar series called Project Safe, the impact of chemicals in our food and our environment. And over the last year, we have been fortunate to have great speakers discuss all aspects of these topics, all the way from understanding the chemicals in food and the environment to the impact that they have not only in food allergies, but also in human health and chronic disorders. Um, and specifically, a lot of the discussions have been almost on the negative impact of chemicals in food and the environment on human health. So today I'm thrilled to pivot from that and have our last discussion with Dr. Sophie Newton from Nestle Health. Uh, because today we're going to talk about more positive impact of chemicals in food that can be used to train our microbiome and our gut health. So very excited to have Dr. Sophie Newton from Nestle Health. Sophie is the global science lead for allergy at Nestle. And as a global science lead in allergy, Dr. Newton selects and promotes scientific and technology discoveries with translational value and facilitate their translation into development opportunities or product concepts or services. Sophie studied molecular and cellular biology at the University of Lyon, France, and she received a PhD in immunology and parasitology from the University of Sciences of Lille, France. Uh, she has worked successfully in immunomodulatory properties of lactic acid, bacteria, and intestinal bowel disease. And one of Dr. Newton's focus is the renovation and innovation of extensively hydrolyzed and amino acid-based infant formula dedicated to infants allergic to cow's milk. So Sophie is an expert on this field, especially when it comes to understanding and utilizing chemicals to promote a healthy gut, which is so important, as all of you know, so important to for human health and also for preventing food allergies if you treat them hopefully at the right time. A very important topic that converges with our goals in food allergies. So before I pass it on to Sophie and start, I just want to remind everyone that we have the Q&A box at the bottom of the screen, and we're going to have about 10 minutes after Sophie's presentation to go through to questions. So Sophie, it's really awesome to have you here. Great to see you again, and very excited to have the opportunity to learn more about your work.
Thank you very much, Carlos. Thank you for the invitation. Happy to share with you today, indeed, a positive outcome of how we could use food bioactives actually to promote a healthy immune gut in early life. And I will start with a few slides of background and then show you one typical example of the latest news in infant formula. So if we start with this graph, just to show you the interconnection between microbiome and immune maturation in early life. So if you look here, for example, so we have the 3 first years of a child, and it's well known now that there are a sequential development of this microbiome, starting with a very low diversity that increase with time. And this low diversity is accompanied by a higher level of bifidobacteria, especially in breastfed infants, which lowers afterwards. And at almost 3 years of old, it's acknowledged that the child has get a more stable microbiome, and that it will stay there for during the adult life. And what is quite impressive, if you look at these curves on immune maturation, you see that it fits quite well with the curve on, for example, diversity. And what is known now is that there is an intricate interaction between the microbiome development and the immune maturation, which is, of course, very important because it helps in the early infancy to protect for infection. That's what you have here, with a higher susceptibility at infection until the immune system is really set up, then decrease after the immune system is set up. And you see here an important point that very early life, so in the few first months, the susceptibility to infection is quite low. And this is due basically to breast milk antibodies, so breast milk bioactives. And I will show you that in 1 second in the second slide, because we always think of human milk as providing the nutrition that a baby requires. And of course, it's the best way to feed a baby. It's made for nurturing the baby. But human milk also contains a lot of bioactives all around here, including growth factors, but also long chain fatty acids, immunoglobulins, as just mentioned, which are very important for the protection of the baby, but also specific carbohydrates, and I will come back to that later. And again, all these bioactives are important factors that, on the top of bringing the macronutrient that needs the infant to grow properly, these bioactives are very important for the gut microbiome, immune development, and also mucosal barrier development. So basically the gut health that should start in a healthy way. So not all infants, unfortunately, can be breastfed and can receive this gold standard food. And that's why infant formula have been developed basically to compensate the lack of breast milk when it's needed. And typically, in a century of history, infant formula have gone from a simple combination of cow's milk with basically water. And then there have been a lot of different technological processes that made infant formula a very sophisticated product, going from how to produce lactose-free components, going to hydrolyzed proteins, amino acid proteins, and so on, to address all the needs of the different types of infants. And one of the major milestones in the modification of the infant formula has been the incorporation of bioactive nutrient ingredients such as iron, nucleotides, as mentioned before, long-chain polyunsaturated fatty acids, probiotics, prebiotics, and so on, just to not only bring the growth, the macronutrients needed for the growth of the baby, but to ensure as well that we can provide the function of the breast milk, meaning the bioactives that are important for this early development of the gut and the immune system. And typically, one of the latest and most important improvement in infant formula have been the ability to include or to add oligosaccharides that are similar to human milk into this infant formula. So what are these human milk oligosaccharides? Basically, they are the third largest solid component in breast milk after lipids and lactose. And if you look at this scheme here, you see that HMO, so human milk oligosaccharides, are in higher content than even proteins in breast milk. So it should be that they have a role there to play. And basically, not going into the details, but they are composed of different monosaccharides that are branched together in more or less complex structures. And there is around 200 HMO that have been identified so far in breast milk. So there is a huge diversity and a huge quantity of these oligosaccharides in breast milk. And what is also important to notice is that the amount and the diversity, the variety of the HMOs is unique to each individual mothers and linked to different factors, like, for example, duration of pregnancy. And typically, we know that infants that are early term do not receive the same HMO as an infant that is born at the right time. So what is specific or interesting in this HMO is, so as mentioned before, that's a mix of carbohydrates. And what is very interesting is that they are not there to feed the baby, but they are there to have a specific function. They are non-digestible, so the human gut does not contain the enzyme that can digest these HMOs, which means that they arrive in the intestine as intact carbohydrates, and then they are metabolized by the microbiome or excreted. So they are bioactive compounds, and one of the primary role of HMOs is actually to support the gut microbiota. So if we go a bit deeper into the functions of HMO, you see on this cartoon, so it's very busy, but I will not go into all the details, but I like it because it's kind of a nice summary of all the function or roles of HMO. So you have these HMOs here at the bottom of the slide. The first discovered role of HMO was the prebiotic effect, meaning that they promote the growth of beneficial bacteria, including and much more specifically than other bacteria, the bifidobacteria. And if you remember well, that's one of the first bacteria that colonize the gut in early infancy. And that's also one of the major difference between a breastfed infant and an infant formula fed infants. Breastfed infants have much more bifidobacteria in their early life gut than the infant formula fed infants. So what is important to mention is that then these bifidobacteria, using the human milk oligosaccharides, can afterwards produce some metabolites that have been related to health benefits, like short-chain fatty acids, for example, which have been involved in gut barrier function. They have anti-inflammatory effect and so on. So it's really the way to promote the beneficial bacteria that leads to the good metabolites to be produced, and also induce the right cross-feeding. So you know that microbiota is interactions between different bacteria. So having the right ones basically can help decreasing the potential pathogen ones. So it's really an equilibrium that is promoted by the human milk oligosaccharides. On the top of it, people have shown that human milk oligosaccharides could also kind of induce a direct protection against pathogens by binding these pathogens or act as a decoy receptor to avoid the penetration of the pathogens in the gut wall. So they cannot adhere to the surface via their receptors. They the pathogen bind rather the HMO than the receptor on the epithelial cells and then cannot enter and induce the infection. HMO also help educate the developing immune system, and that's mainly in vitro studies that have shown anti-inflammatory effects of these HMO. And finally, via different ways, they can strengthen the gut barrier, either through the metabolites produced, as mentioned before, or directly by inducing specific genes, for example, for tight junction, where they can really strengthen the gut barrier. So that's basically the four different major roles of the HMOs that have been described and proposed. So now how we translate this knowledge to what we can do as an application for infant formula. So basically we know that HMOs are good in breast milk. Can we use this knowledge and can we mimic in infant formula what's in breast milk? And there is one big shift in the last 10 years. Technology, actually, it's technology driven, that enabled the chemical, from chemical synthesis of these HMOs to a biotechnological manufacturing that made the HMO bioactives accessible in terms of those quantities. So we could produce a lot, and also at prices that are within the range for commercial application, because we need to consider this for translation into reality. That's nice to know what HMO are doing, but we need to be able to apply it. So based on the lot of research, the technology has been shown now and approved by regulatory authorities such as EFSA and FDA to be used for infants, and they have obtained the GRAS status, meaning generally recognized as safe. So based on that point, the different points, quantities, price, and safety and regulatory assessment, we could then start trying to incorporate these new bioactives into infant formula. And this is easy to say, not easy to do. And that's true for any new bioactives that we need to include. The first step is, of course, to identify the supplier that could bring the right quality and at the right price, basically, to be used. Then, of course, the food safety evaluation is a key step. We need to ensure that including these new bioactives into the process of making the infant formula is safe. And of course, we need to adapt the manufacturing process as well to ensure that these new compounds are still bioactive at the end of the process, because the processing induce heating, induce hydrolysis for some of the infant formula. And so we need really to ensure that what we put in at the beginning is still okay at the end and can do the function that we request them to do. And of course, last piece of work, which is very important, is the clinical trial to demonstrate both the safety in the different target population and the efficacy, of course. And just to give you as an example, for example, for specialized infant formula, so I mean amino acid formula and extensively hydrolyzed formula, which are the ones that are used to feed the cow's milk allergic infants, we need, of course, on the top of it to show that the bioactive that we add on this formula lead to still an hypoallergenic formula, so that we do not introduce any traces of cow's milk or that could induce any reaction to this target population. So we really need to adapt to the target population and to ensure absence of traces of milk protein, low lactose content, and so on, that is required for this category of products. So that's, in a summary, all the work that needs to do from the identification of a potential bioactive that could be helpful for the kids having infant formula and not being breastfed, to the reality of the commercialization of infant formula containing these bioactives. So I will now go to show you some examples of clinical trials that have been run to actually show what the effect of this HMO when they were added to an infant formula. So here we start with a standard infant formula, and what we could show is that you see here we had a breastfed group, a control group, so that's a standard formula, and a standard formula plus HMO, that's the test group. And what we looked at is the microbiota and this is in 3-month-old infants. And you see that compared to breastfed, and here in red you have the fecal community type that contains high bifidobacteria. So that's expected. We see high bifidobacteria in these breastfed infants, while in the infant formula you have much less. And what you see here, that when we add HMO, you can increase again these bifidobacteria, which are known to be very important for the early development of the kids, and which is one of the functions that is acknowledged for HMO is this bifidogenic effect. So we could show in clinical trial that it works. Basically, adding HMO can help providing a better microbiome composition at the start of the life. What we looked as well was infections. Again, if you have a better microbiome at the beginning of life, you also provide a better way to have a better immune maturation. And that's what we showed here, comparing the control formula here with the test formula. And here it's parent-reported lower respiratory tract. And you see that you have an important decrease in this lower respiratory tract infection in the group that have had the HMO. So that's one of the first clinical trials that show that adding this HMO into an infant formula can be translated into benefits for the kids. Now, since we discuss allergy and food allergy, one of the questions was, does this HMO also have could have an important impact on cow's milk protein allergic infants? And of course, this is based on the fact that cow's milk is an immune-mediated disease which is linked to dysbiosis, increase of gut permeability, Th1, Th2 imbalance, and that these kids have a higher risk of developing infection. So we thought that potentially HMO could be of important use for or function for these infants. So we did similar studies comparing control that's on the left side with the test formula, so same formula with HMO, and here we are talking extensively hydrolyzed formula. So not going into too many details, we could show again that we see a difference going for on the right side of having more bifidobacteria when we added HMOs. And similarly to what was shown in healthy infants, we could demonstrate that the implementation of HMOs in these infants with cow's milk protein allergy could decrease respiratory infections and also ear infections. And just one last slide of data. This was done on amino acid based formula, but I thought that it was quite interesting to see here that. So again, we have here amino acid based formula with HMOs that were tested in infants with cow's milk protein allergy. And you see here the enrichment in the HMO utilizing bacteria. So as mentioned before, not all bacteria are able to use this HMO to metabolize them and induce these health related metabolites. But you see here an increase in this HMO utilizing bacteria, meaning that we can again modulate with HMO the microbiota composition, and potentially that what you have on the right side generate an increase in short-chain fatty acids a long time when the infants are fed with these HMOs. So just to conclude and leave some time for some question if you have any. So what I try to convey as a message is that the developing gut microbiome in infancy plays a key role in shaping the immune system. So that's really the target timing where we need to ensure for infants that they have the right development of the gut microbiome. The human milk, of course, contains all the needed bioactive components on the top of the macronutrient, and they have an impact on microbiome and immune development. And the aim of research on infant formula is really to constantly improve this infant formula with the aim of course, providing the most optimal nutrition, but also as much as possible mimic what's good in the breast milk and adds the bioactives to the infants that cannot be breastfed, so they can beneficiate from the effect of these bioactives. One of the latest development, as mentioned, is the supplementation of this infant formula with HMO. So that's a big change in the infant formula field. And while they were discovered a long time ago, as mentioned recently, the technology advances make them available to be used in quantities and at prices that allow this commercial application, specifically in infant formula. And now we have a lot of trials, we and others, showing that HMOs are safe for infant and that they have an impact specifically in supporting the microbiome and the immune development of infants. And that's it. So thanks for listening and happy to hear any comments or question if you have any. Thank you very much for listening.
Excellent, excellent presentation, Sophie. Thank you so
Thank
much.
you.
It's really great to see this other side of chemicals in food that can impact human health. Um, Before we get into Q&As, everyone always asks us, how can they help? Donate, host an event, follow us on social media. Here's a simple way to connect with us. You can use this QR code. Uh, and so let's go to question and answer. I'll start. So Sophie, really exciting, and you show the complexity and the diversity of oligosaccharides, human milk oligosaccharides, and the impact, clear impact it's having on the repopulation of the gut microbiome. It's interesting that all these oligosaccharides have different structures from a chemistry point of view. Chemic uh, um, carbohydrates are very labile, right? And they can break in acid. And so I was wondering, is the effect that you see with the human milk oligosaccharides, is it more of because of when they get hydrolyzed, there's the individual components now can serve as building blocks, or is it something in particular about their structure that now can drive some specific biology? And if so, are they protected somehow with other proteins and other things when they go through your stomach?
So yeah, so basically we believe that it's really the structure. And I did not go into the details, but it's really complex structure, which is very unique to these human milk oligosaccharides. Totally different, for example, from FOS and GOS that are well-known prebiotics. And it's because they have specific branched that link the different monosaccharides together. So you have some that are quite linear, some that are very structured ones, more or less big. And basically, as mentioned, the human gut do not have the enzyme that can separate the different monosaccharides. So they arrive as the structured oligosaccharides in the gut, and they are taken up by bacteria. And that's the bacteria that can use it, actually. So it's really the way it's working. What is still not fully understood is why we have so many, why the Mother Nature has made 200, that's what we know today, different structures. So we know now that they can be in different, I would say, families. Some that are more acidic, some are neutral, some are fucosylated. So they probably have different functions because they have different structures. There's still a lot to do, research to do on that. What we know, for example, is that sialylated ones are more linked to potentially cognitive function than the other ones, linked to this sialic acid that they have on the top of it. But yeah, very complex. We do not understand it all. The idea behind today is that we probably need to diversify to have the full function that can be brought by HMOs because again they have different structure. But what we don't know if we have, so we have seen, for example, the examples I've shown is two different HMOs only, and we can already see some differences. So yeah, there are probably redundancies between the different HMOs, but a lot of research to come.
That is
We
great.
understand it. It's amazing. Yeah.
I know we only have one question. I'll just take one more question, given the time. Uh, and it is more food allergy specific. If an infant was exclusively breastfed, what other factors could have impacted their microbiota to lead to food allergies at 6 months of age upon first exposure to an allergen? Thank you, Hannah, for that question. Any thoughts on that?
Yeah, that's a big topic, and I don't think anyone had the the answer for that. Of course, it has been shown that breastfed is the best thing you can do for a child. That's the yeah the gold standard way of feeding your baby. Now I think and you had some of these discussions in the previous sessions. Environment can play a role, immune dysregulation could play a role. So uh, yeah, I think that no one had the right answer to this question. It should be something that triggered wrong or not optimal immune maturation during that time. Although I mentioned microbiota is important, and with breastfeeding, you should be able to provide and to develop the healthiest microbiota. But as yeah, always in biology, I think that's one piece of the puzzle, but probably there are other interacting factors that, like yeah, again, environment, maybe chemicals in the air. We don't know basically what would happen. But
Great.
maybe Carlos, you have other thoughts on that, huh? Because it's I know that it's also a topic that you are
It,
really
it,
like. So
there are many
yeah.
factors, microbiota being one of them, but there are many factors. And, to your point, the most important thing is, research to be done, a lot of questions to be answered. That's part of why we're here, trying to answer all of these questions. So I know we're out of time, but first, Sophie, I want to thank you for taking the time for great talk, and especially with ending this seminar series in such a high note of the positive impact of chemicals in human health. And I want to thank our audience for sticking with us throughout this seminar series over the last 12 months. Has been an exciting topic. I feel that we've learned a lot, and what are the impact of chemicals in the food and the environment that may be associated with so many of the many chronic disorders and food allergy cases that are developing, and also ways that we can leverage some of those chemicals from nature to help us protect us from some of these disorders. So thank you, everyone. Stay tuned. We're going to start another seminar series, very different next year, which is going to be focused on clinical studies in food allergies. So I really hope all of you can join us, and thank for being with us. So thank you, Sophie.
Thank you very much for the invitation. Thank you.